Well, good afternoon. I'm delighted to see everybody here for what is, at the same time, the first President's Lecture Series of the academic year and the last of the calendar year. And whether you look at it as the beginning or the end, I'm sure it'll be a treat. I am really delighted that we're going to be hearing shortly from Todd Roberts, who's Associate Professor of Neurosciences and also an investigator in the Peter O'Donnell Brain Institute. Todd joined us in 2013 to pursue his interest in understanding how memory is laid down and using what will not be a secret by now to anybody who's here, songbirds to understand auditory memory. This has a great relevance in helping us understand how we develop language ultimately and insights into that, and also into processes that are almost certainly going to be relevant to a lot of important medical challenges, neurodevelopmental ones, autism spectrum disease, and more. And before I give my further introduction of Todd, I'd like to make note for those who may have come in this side and not been aware that over on that side of the Gooch, we have a table there for our newest business-related group, BRGs. It's the Equally Able Alliance, BRG. We encourage you to take the opportunity to visit and understand more about it. It's a group of like-minded members of our UT Southwestern community focused on how to be sure that we're providing the opportunities for people of all manner of ability to contribute and achieve their full potential. There is obvious resonance here for understanding some of the neural circuits, which are part of which help us do just that. So let me, as I said, introduce Todd. As I mentioned, associate professor who joined us in 2013. That was after completing his doctoral work in neuro and cognitive science at the University of Maryland, and then his postdoctoral work at Duke University. And he came here to pursue the work we're going to hear about, and over the course of time has come to use a variety of techniques to really gain insight into these really fundamental and important processes. And that includes, coming up to present day and recent studies, applications of artificial intelligence as well. And so I'd like to note before handing the podium over to him that his creativity, innovation have been recognized. He was the recipient of the NIH Director's Transformative Research Award in 2021, which recognizes exceptionally innovative research with the potential for extraordinary impact. I think we'll have an opportunity to see just why he received that award shortly. So if I can, I'll welcome Todd to the podium.
Okay, thank you, Dr. Podolsky, for that introduction, and thank all of you for being here today, and also the people that are attending over Zoom. It's my distinct pleasure to talk to you about some of the work that we've been doing and why we're doing this type of research. So as Dan mentioned, we study songbirds, and so the first question is, why do we study these little dinosaurs? Why are we interested in studying songbirds? And this is the species that we primarily study here. These are zebra finches. There's a female zebra finch in the foreground. In the background is a male zebra finch. The male zebra finch has this orange cheek patch and also stripes across his chest, and that's why they're referred to as zebra finches. And the reason that we study them is related to the fundamental interests of the lab, and that is trying to understand how we learn behaviors, complex behaviors, through imitation. So many of the behaviors that we learn that get us through our daily lives and allow us to socially interact with one another were learned early in development through imitation. Classic examples are, of course, what I'm doing now, speech and language. This is a primary mode that we use to communicate with one another. We learn speech and language through imitation by listening to our parents or other individuals while we're young, and then learning how to imitate those behaviors and develop them. And other common behaviors that we learn through imitation are like playing musical instruments or playing sports, at least at the initiation of learning these types of behaviors. We're often engaged in observing someone else who's more experienced than us do this, and then trying to copy what they're doing. And so this involves forming memories of how people perform certain behaviors, and then learning how to mimic those, or learning how to adjust our own performances based on those memories. And songbirds, like us, have to do a lot of learning when they're young. So young songbirds have to engage in a type of imitation in order to learn how to produce their songs. They do this during developmental sensitive periods. And in part, what they're trying to do is learn a behavior that's going to be critical for them to be able to socially interact with one another when they're older, and also to be able to attract and keep a mate, and to have breeding success. So in this first video, what I'm going to show you is how this juvenile learning first takes shape. So there's adult male is here, and this is a juvenile. When I play the video, their positions are going to change. But what you could see is when a young bird is being tutored by his father will sing to him, peck at him to make him pay attention, and then he'll sing. Okay, so by any standards, this isn't the most beautiful song. It's repetitive and a little bit chirpy, but this is how young birds can first form memories of their dad's song, and then eventually use those memories to guide imitation of the behavior. And so how does this learning unfold? So for zebra finches, this learning occurs during the first 3 months of life, during a sensitive period, and it starts out with a phase of sensory learning where the young birds form a memory, and this is a lifelong memory of their father's song or the song of another adult male that tutors them, and they have to memorize the temporal and spectral properties of the behavior. And then starting about the second month of life, they start to practice their song, and then will practice this extensively until they're about 90 days old, until they get a copy of the song that is near perfect to what their father was singing. And then in adulthood, they'll use this song to try to attract a female mate. And in this species, they will mate for life, and the male bird will sing his song for the rest of his life and use that song to maintain these bonds between the male and the female. And there's obvious parallels here between what these young birds are doing and what we do when we're either teaching our kids speech and language. We do this instinctively, like seen here in the video. Anybody that has kids knows how much time they spend speaking to their infants in kind of exaggerated tones and making facial expressions to get them interested in paying attention to them. And then as infants and young children, we go through an extensive period of time where we're learning, going through babbling and stepwise acquisition of individual words and stringing words together. And birds do a similar type of thing, where they go through an extended period of time where they're learning individual syllables that dad sings, and then learning how to string them together in a complicated way. And I'll show you a little bit more about that in a minute. And then in adulthood, the zebra finches at least use this song to attract a mate, as I said. And so in this video, what you're going to see is there's a male bird here, and behind this glass is a female bird. When we pass current through the glass, it becomes clear, and then he'll be able to see a female bird, and you can see how he behaves. So again, now I think you could see, how stimulated the male is to try to produce a song and this courtship behavior to the female, and again, driving home the saliency of how important this behavior is for the young male bird to acquire during development. So just to recap this, young birds need minimal experience to acquire lifelong memories of their father's song or the song of another bird. They practice their song extensively in order to get it right. In this species, they'll practice well over 100,000 times during development in order to master their song. Just in a chamber alone, they might sing 3,000 times a day, just practicing. And the adult song is stable for years, and it requires auditory feedback in order to maintain it. And again, this is very similar to how we maintain our speech. We need auditory feedback in order to maintain our speech throughout our life. Okay, so what does song look like? So on the bottom, you could see an amplitude envelope of the bird's song, and above that, you could see a spectrogram. And so this is a visual representation of song that shows frequencies on the y-axis and time on the x-axis, and the color coding shows you the power across these different frequencies. And this is the equivalent of what you just heard that bird sing. And when we look at this to quantify it, to start to understand it, we break it up into its component pieces. And so first we break it up into syllables. So you could see this bird sings what we refer to as introductory notes with these eyes here. And then he sings syllables A, B, C, and D. And then he just repeats them. A, B, C, and D, A, B, C, and D, A, B, C, and D. And so we call that repeated structure a motif. And so that's really the learned unit that the bird has to learn from his dad. And then he can put that together and repeat it over and over again. And in these repetitions, we call that whole behavior a song bout. And females are judging him on how well he produces this entire song bout. Okay, so now that you know a little bit about spectrograms, let's take a visual representation of what this learning process looks like. So on the top spectrogram, you could see a motif of a father bird. And so he's got like 5 syllables here, and you can tell that they have different spectral features to them. They look different from one another, but they also are sung in a certain order, and that order is stereotyped. And then on the bottom is the song of his son, one of his sons when he's really young. And what I'm going to play back to you now is several thousand versions of what the son produces as he grows up. And so you could see how his song changes from this really noisy kind of babbling, unstereotyped behavior to something that's quite precise. Okay, so you just need to get the gestalt of this. So it doesn't look like there's really any pattern to what he's doing yet. And then now you could start seeing that he's producing something that looks like this syllable, or this group of syllables. Like he's doing it here, but he's also singing at the front of the song, at the back of the song. And then he starts producing something that looks like this syllable as well, but it's kind of all over the place when he's producing it. So he's singing things in the wrong order. And now he seems to start to get the syntax or the grammar of the song. He starts producing things in the right order. And now he's getting closer and closer to adulthood. And now you could see it starts to really lock in. And you can see how precise this is. Every time he sings it, he's singing the exact same thing. And it's that precision in this behavior that's quite remarkable. This is equivalent to a professional operatic singer, where the precision of how someone can how this bird can produce the exact same thing each time. And part of that is that this type of precision is something that female birds really care about. Okay, so what can we do with this type of behavior? Well, there's several questions that we can ask that are important for neurobiology. How are memories of the social experience used to guide this imitation? How do they form these memories? How does the brain, is it able to detect errors in performance and then correct those errors? And lastly, how can we use this type of model system to study neurodevelopmental disorders? There are a lot of neurodevelopmental disorders that have strong impact on the development of these behaviors that I think are some of the most complex behaviors that we produce, like learning speech and language, like learning social behaviors from one another, imitating each other and learning how to act in certain situations. All of these things we develop and learn through social experiences and observation of other people in our environment. And I think this provides kind of a rudimentary model system where we start asking questions about how the brain does these types of computations. Okay, so why is song learned? This song could just be innately specified. Most animals, their vocalizations are innately specified. Non-human primates have a lot of vocalizations that they produce, but those are all innately specified. We're the only primate that learns their vocalizations in the way that we do, by imitation. So the answer is that females are really driving this in this species and the evolution of song more generally in songbirds, and there's about 5,000 species of songbirds. So if you look at an imitated song on this top row here, what the bird ends up producing is something that is very stereotyped and has complex syllables in it. If you raise a bird where he never has access to an adult tutor, a bird that's going to sing to him and teach him what he should be producing, he'll end up improvising his own song. So he'll just make it up. And when he does that, he ends up with a song that is highly variable and has really simple syllables to it. And those songs are not preferred by females. So if a female has a choice between mating with a bird that has this song or this song, she'll always choose the bird that has the imitated song. And so we wanted to understand, what are the deeper aspects of this behavior that females are listening for? It's not just stereotypy or complexity, or what does complexity actually look like? And so recently we developed some computational approaches to look at the song and the song syllables at large scale. And we can do this by first being able to parse hundreds and hundreds of thousands of song syllables from individual birds from these audio recordings. And then we can take the spectrograms of those and then we can flatten them and then project these into low dimensional space. So this is a really high dimensional data set, but we can project it into lower dimensional space. And this is a UMAP plot, but there's several ways that you can present this. And in this plot, what we can do is look at the individual syllables that birds produce. So each one of these clusters is roughly 2,000 syllables from one individual bird. And they clustered tightly together because the behavior is so precise. So each time the bird produces that syllable, it sounds very similar to itself. And then we can look at where these syllables from one bird that are circled here are distributed in this space. And then when we do this, when we're comparing birds that learned their songs and birds that were raised without access to an adult tutor and had to make up their songs. So again, songs that females really prefer versus songs that they don't prefer. And we look at how these distribute in this lower dimensional space. And this is looking at about 500,000 song syllables from a group of 50 birds. What we found was, at least one of the things that we found, is that the learned songs span larger trajectories in this low-dimensional space than songs that were improvised or the bird had to make up. And we see this consistently across our birds. And then we wanted to ask, is this biologically relevant, or is this some just artifact that we happen to see and it doesn't really mean anything to the birds? And so we asked whether or not females actually prefer these songs that span longer trajectories in space. And so we made versions of synthetic songs that all came from imitated syllables, but then just span different distances in these low-dimensional spaces. And then we played these back to females in free flight arenas where they can go to where we're playing either the longer path length song or the shorter path length song. And we just asked them what they seem to prefer, place preference test. And what we found is that females strongly and reliably prefer these songs that span longer distances in these low dimensional spaces, which suggests that not only can they detect the differences in these songs, but they also show very strong preferences for one over the other. And so females finches strongly prefer longer path length songs. And then I'm not going to show you here, but we went on and studied whether or not this could actually be a marker of fitness in some way by looking at whether or not this is a difficult thing for males to do. Do males have a harder time to imitate songs that span longer trajectories, or do they have a harder time learning ones that have shorter trajectories, or does it not matter? And what we found is that male birds, young male birds, have a harder time learning these longer trajectory songs, suggesting that this could actually be an honest marker of fitness that females are honing in on, because it tells them something about the quality of the male, that the male has been able to, during development, spend a lot of time learning this behavior, and that maybe it has some reflection on the intelligence or some other aspect of the male, or his environment and his ability to feed himself and do other things instead of just singing and practicing song all day. There's a lot of things that are involved in survival other than taking 5 hours a day to practice your song. So then we wanted to see whether or not we can understand what it is that is associated with these longer trajectory spaces. And we did lots of analysis of the spectral features and temporal features, looking at correlations across these different spaces, low-dimensional spaces. And what we ended up finding is that the trajectory of syllables really reflects the spectral and temporal distinctiveness of the individual syllables that make up a bird song, suggesting that this is a nice marker of complexity, and that this marker of complexity has evaded previous research. So, along with this research, recently we've been using a lot of artificial intelligence approaches to develop methods to more fully and automated ways of analyzing this type of behavior, vocal behavior, to deeply characterize it, deeply characterize the developmental process, and also deficits in that developmental process, which is really critically important if we want to use this model system to study neurodevelopmental disorders. We need to have a really good handle on the behavior, because ultimately that's going to be the readout for whether or not there's disruptions in learning. Okay, so what about the brain circuits for this? If we want to study neurodevelopmental disorders, we have to understand what's going on in the brain and what areas of the brain are important for this type of behavior. So early anatomists this is a sagittal section of a zebra finch brain, and early anatomists realized that when you look at the brain of a zebra finch, or really any songbird, that there's these prominent areas in the forebrain that stick out. They look like these hypertrophied regions. This is a myelin stain, but when they compared this to the brain of something like a pigeon or a chicken, and birds that don't learn their vocalizations, they didn't see these regions. And they wanted to understand whether or not these regions are important for song. And early anatomists would then go in and lesion these areas, and they found that indeed that they are critical for the bird to be able to produce its song. Nowadays, we have a much more complex view of the neural circuits that are involved in song. This is a 3D image of a songbird brain generated in our lab, an image at the whole brain microscopy facility. And over the years, we and many other research groups have mapped in detail the neural circuits that are associated with singing. And one of the things that we've come to understand is that this area, HVC, serves as a central hub for song. And so this area is needed for the birds to be able to produce a song. It also seems like it's needed for the birds to learn their songs. And so it's receiving auditory inputs, and also the core of the outputs that form pathways for producing the song, and also pathways that are involved in practicing and changing the behavior over development. Okay, so with that, let's step back and start getting to some of the questions about, what can we learn about the neural circuits here. And the first thing we wanted to do is identify in detail whether or not there really is a neural circuit that's necessary for song. And as I mentioned a minute ago, people have found that lesioning this area HVC disrupts song. This is just an experiment from our lab. This is the bird's song. This is a spectrogram, just one that is not in color, before lesion, and then once we lesion, this is what it looks like. And when you do this, the song essentially reverts to the juvenile form of the behavior. So it sounds like the earliest stages of the song learning process after you lesion HVC, and so all the learned components disappear. Now, to do this in a slightly more sophisticated way, we've started now to apply optogenetic methods to manipulate neural circuits in freely behaving birds. Now, optogenetics is a tool in neuroscience that allows us to express channels in neurons that allow you to control the activity of a cell just using light. So certain optogenetic channels, if you shine light on the neurons that are expressing those channels, it causes the neurons to become active, to fire action potentials. Or if you use a different channel, you can suppress the activity of the neurons. So you're either enhancing or suppressing the activity of the neurons. But you can do this with light, you can do it with millisecond precision. And you can do this in freely behaving animals. And so this is an example of us expressing an optogenetic channel named carmine, which is an excitatory channel. So it will strongly depolarize the cells or make the cells become active when we shine light on it. And we could do experiments where we express this channel, and then while the bird is behaving and singing, we could see what happens if we buzz HVC, essentially. And this is an example of one of these experiments. And so this bird's got fiber optics implanted above HVC. And what you're going to see is that he's going to go over to the mirror and he's going to sing to himself. Birds love to have a mirror in their cage because they'll think it's another bird, and they'll sing a lot to it. And then he'll move away, and then when he goes back, we'll optogenetically manipulate HVC while he's singing. Okay, so now this time, we'll optogenetically stimulate him a couple times while he's singing. So it's difficult because the behavior is so fast, but maybe what you heard there is that he started, stopped, and then he started again, stopped, and tried to start again. And we can look at this by doing lots of different things, but one of the ways that we've looked at it is doing respiratory recordings from birds while we're doing these manipulations. And so in order to produce this behavior, they have very stereotyped respiratory patterns that underlie their production of it, just like a singer or we do when we're speaking. We really have to control our inspiration or expiration. And on the top, what you can see here is the respiratory patterns when a bird is just normally singing its song. All of the lines going above this dotted line are expiratory pulses, and that's when he's producing his song. And then when it goes below is when he's taking many breaths in so that he can sing the next moment in time. And hopefully what you can see here is that there's two motifs that the bird sang, and those respiratory patterns are repeated very precisely between those two motifs. Now on the bottom, what you can see is that when we time lock our optogenetic manipulation to this moment in the song, and so you can see the respiratory patterns are really well aligned across many trials, and as soon as we turn the light on, the bird truncates, stops singing, and then with a few milliseconds he starts singing again. And what we see is that when we manipulate this circuit, what the bird is really doing is skipping back, kind of like a scratched record, where you manipulate the circuit and wherever he is in the song, the neural network stops and then starts back over at the beginning, suggesting to us that there's a circuit in HVC that's controlling the pattern generation that's needed for the bird to be able to produce a song. And it only has one starting point. And so whenever you do this manipulation, it has to skip back to where it began. Another way to see this is through these spectrograms, but I'll just point your attention to this lower one where we're repeatedly light stimulating as the bird is singing syllable A, and he repeatedly just goes back to the beginning of syllable A over and over and over again. And another way to see this is if we take many hundreds of these spectrograms and collapse them, and so that's on this axis, so the spectrograms are going across this way, and what you can see is this red line that's scanning down, that's the stimulation times at different points in the bird's song. And so it really doesn't matter where we are in the song. If we stimulate an HVC, it causes the bird to immediately truncate, and then it skips back to the beginning of its song again. And then the last way to see this is through this type of plot, where all of the black dots are the stimulation times, the gray X's are when the bird truncates, and then the orange dots are when the bird restarts singing its song again. Suggesting that we've really tapped into a pattern generation circuit that's essential for the bird to be able to produce this behavior. Okay, so what we are doing now in terms of using these optogenetic approaches to better define this circuit is going in more detail into what are the neurons in HVC. So it turns out this just isn't one population of neurons in this area of the brain. There's multiple intermingled populations, and we're using these light-based approach where we can excite neurons and combine them with electrophysiology where we can record from other neurons to in detail map who's talking to who in these networks. So we can figure out in this complicated pattern generation network, what are the neurons that are controlling different aspects of the behavior and how are they connected with one another. In addition to that, we're also mapping for the first time the long-range connections in these pathways. And so how are all of these pathways that are projecting into HVC, who are they talking to in this network? And this has only really been made possible by a lot of work in our group where we've extended the use of viruses in this system, enabling us to express these optogenetic channels and really map the circuits in detail. In addition to this, we're also using optical approaches where instead of just shining lights through an LED and hitting everything in the circuit, we're using sculpted light and holographic patterns of light where we can actually guide light to individual neurons in the network. And through a 2-photon microscope, we can go and see the different populations of neurons and then know exactly which neurons we want to manipulate and ultimately try to understand how the different populations are encoding for different aspects of the behavior that they're trying to control. Okay, so after understanding that there is a neural network now that controls the generation of the bird's song, we wanted to understand whether or not this pattern generation network, how is it taught what to do? How is it taught what pattern it should be producing to underlie each bird's song? Because not all our birds have the same song. You learn the song of your father, but we've got lots of dads in our colony, and we can dissect who belongs to who based on their song. And so we wanted to figure out whether or not this pattern generation network is also involved in the initial learning through auditory experiences when birds are young. And I started this type of research years ago when I was still a postdoc. And what I found when I was still a postdoc, by using longitudinal imaging approaches where we can track synapses of individual neurons in HVC over the learning process through development and see how plastic these neurons are, what their synaptic connections are, whether or not they're strengthening, et cetera. What we found is synaptic plasticity in HVC is needed for birds to learn the song of their tutor. And so this area that's not just involved in producing the behavior when they're adults, when they're young, is also essential for learning the behavior. And one experiment that we were able to do that kind of solidified this idea was where we can manipulate the neurons in HVC, again using optogenetics, but do it during social learning. So when a young bird is learning from its dad. And so in this experiment, instead of manipulating the bird that's singing, we're manipulating the tutor, the young bird, the juvenile that's trying to learn from the dad. And so what we have is the young bird here, the pupil. He's got optogenetic channels expressing in HVC and then fiber optics over HVC. And then when the dad sings, which is this guy, you'll see what happens. So the idea here is that if auditory information from listening to the father is flowing into HVC and important for the neurons in HVC to learn from those experiences, by optogenetically manipulating their activity and getting them to fire action potentials in a way that's not associated with what the input structures might be telling them to fire, that it should disrupt learning. And that is exactly what we see. So if we optogenetically stimulate while the bird is listening to their dad, and we do this for a few days while they're being taught, and then we just raise them to adulthood, we see that these birds don't learn anything from their father's song. It's as if they never had the experience. While other birds, control birds, that underwent the exact same experiment, except for we just optogenetically manipulated activity in HVC at moments in time when the dad wasn't singing, so they still had the same amount of optogenetic manipulations. Those birds went on to learn perfectly fine, telling us that in real time, as a bird is having these social experiences with their father, auditory information is flowing into this network HVC, and that is critical for the birds to be able to form these long-term memories that they use to guide vocal learning. Okay, so using this as a backdrop, the next idea was to try to figure out, so we know auditory information is important, it's flowing in there in real time, and it tells the bird something about what it should be learning, what its song should sound like, what its syllables should sound like when it's an adult, and they'll use that memory in order to guide changes in their behavior over development. So what we wanted to do was then see if we could just step directly into that loop and manipulate the auditory inputs to HVC directly using optogenetics. And so what we did, instead of manipulating inside of this network and these neurons, we expressed these channels in an area that we thought would be an important source of auditory information to HVC, and we had some evidence could be important for learning. And so we express optogenetic channels in this area NIF, and then shine the light over their terminals in HVC at the end of their pathway in this network. And we show that this type of experiment works when we do neural recordings in HVC and we shine light over HVC. We show that we can control the activity in that network. And then we designed an experiment that we refer to as opto-tutoring. And so we take young birds that haven't had any experience with a tutor, they haven't had social interactions with a tutor, so they're tutor naive, and then we give them opto-tutoring experiences where we're manipulating the activity in HVC using different patterns of light. So driving activity in this network with different temporal patterns of light to see if that guides learning in some way. And in the first experiment, we gave the birds packets of light that were 50 milliseconds long, and we tutored the birds with these packets of light for an hour and a half each day, 300 trials each session. And again, this is a bird that's all by itself. There aren't other birds around it. It doesn't have an adult bird to learn from. And then after we do this for 5 days, we then remove the fiber optics, and then we just let the bird naturally grow up and see what it learns in adulthood, what its song looks like. And so just as a quick reminder, this is what a normal zebra finch song sounds like. And then the next one is the song of a zebra finch that was just raised without a song tutor. Maybe what you can hear is that song seems a little simpler and also slower. And then this is the song of one of our birds that we raised with this opto tutoring with 50 millisecond pulses of light. Hopefully what you hear there is that it sounds like the bird is trilling in some way. It's got this very fast song that has components that are repeated over and over again at a very fast rate. There's lots of species of songbirds that actually do trills, incorporate trills into their behavior. Zebra finches don't do that, and so this is very odd behavior for a zebra finch, and very unlike the behavior that you see in birds that are raised without having experience of a tutor. And quickly, when we quantify this over a number of birds, what we see is reliably that these birds that are opto-tutored 50 millisecond pulses of light learn syllables that are much shorter and they string them together much faster. And if we look at the duration overall of all of the syllables that these birds learn, their peak duration is very close to this 50 millisecond pulse packet. So it's 62 milliseconds of light. So suggesting that we're actually influencing the memory that is used to guide learning in this species in young birds. Now, in order to really know whether or not we're doing this in the way that we think we're doing this, of course, we have to do another experiment. We have to move the birds in another direction. And so not just 50 millisecond pulses of light, we then have to provide them a memory or an artificial memory of a long song syllable that they should be learning. And so we did this with 300 millisecond pulses of light. Again, much longer than song syllables zebra finches usually produce. And this is the syllable of one of these birds that was trained this way. We can listen to this real quick. So to us, listening to this stuff for years and years and years, this is totally bizarre. Birds never do this, right? Like this, we didn't believe this at first, and I told the graduate student that was working on this, like, you really need to know that this is the thing that this bird is using as his song, and not just some other vocalization that he's producing because, some weird thing that happened in the brain that we don't know what's going on. And so we put birds that had these totally abnormal songs in with female birds to see whether or not they would actually try to court female birds using these completely bizarre songs. So there's the male. And we put the female in. And so it's not just some call or some other thing that they're producing. This looks like the courtship song that they're using, the thing that they were taught when they were young, using these manipulations of neural circuits to implant a memory that then they used to guide how they were going to learn their behavior over the next 100,000 times when they were practicing it and over 2 months of development. Okay, with all of this in hand, then we wanted to see whether or not, now that we know something about these neural circuits, whether or not we can study a gene that's associated with neurodevelopmental disorders and see how it impacts this type of learning in these neural circuits. And autism spectrum disorders is a class of disorders that are associated with different genes. And a lot of autism spectrum disorders, we don't know exactly what's causing it. But there are some that are pretty well tied to certain genes. And one of the ones that we've been focusing on is a gene called FOXP1, which is a transcription factor. And we're focused on FOXP1 because kids that have disruptions in FOXP1 have very impacted speech and language development. Essentially, they don't develop speech and language. And we thought that this could be a gene, if we can disrupt it in these circuits that we know are important for vocal learning, that we might be able to gain some insights in what this could be doing in the computations that are associated with speech and language learning, or at least learning complex imitative behaviors. And so working with Jenna Konopka's lab, we developed methods for knocking down FOXP1 in songbirds. And the first thing we needed to do is know where FOXP1 is expressed in the songbird brain. And it turns out that it's strongly expressed in this area HVC that we have been studying. And what we did is two sets of experiments. We did one set of experiments where we let the birds early in development form a memory of their song tutor. So they've already gone through that part of the learning process, but then we knocked down FOXP1 when they were trying to do the imitation, when they were doing sensory motor learning, listening to themselves and practicing 100,000 times to try to get it right. And then we saw what happened to their behavior. And then in another set of experiments, we knocked down FOXP1 and HVC prior to them having an opportunity to form a memory of their song tutor. So separating these sensory and sensory motor phases of development. And what we found is that these birds that already had a memory of their dad's song, they went on and learned that song perfectly well. They were no different than control birds. And so disrupting FOXP1 seemed to have no effect on this part of the imitation process. But when we knock down FOXP1 in birds before they have an opportunity to form a memory, these birds as adults sang something that did not resemble the tutor at all. And they sang songs that looked very much to our eye like isolate songs, so birds that never had any tutoring experiences. And you could see in these plots, or kind of Markov chain plots, where the thickness of the arrows is telling you the probability of the transitions. In the birds where you knock down FOXP1 after they had a memory, in adulthood, they had very, very high likelihood transitions, just like you expect in a normal bird. The song was very stereotyped. And unlike that bird and that cohort of birds, these birds, their song was all over the place. And so it was highly variable, and they also didn't copy the syllables of their dad. So this tells us that FOXP1 can really disrupt this early memory phase of learning from the social experiences and during these social interactions. And to better understand exactly how these FOXP1 disruptions in song compare to other changes in behavior that we've quantified in the lab, we used a linear discriminant analysis to plot songs of birds that have had different experiences growing up and see how they parse out these different types of singing behavior. And in the lower right, what you could see is many normal zebra finches and where their songs plot out, and so they're all clustered together. On the lower left, you could see the songs of birds that have been raised without the ability to hear themselves sing, and so their songs are highly disrupted because they couldn't use auditory feedback in order to guide any of their learning. And then above on the right is the song of these birds that had to improvise their song, that could hear themselves, but they didn't have a memory of what to do. And we find that the FOXP1 birds overlap with these birds that never had any social experiences, suggesting that when we do these manipulations of FOXP1, it's strongly impacting this initial phase of memory acquisition when the birds are having these extensive social experiences with their dad or another adult male, and they just aren't able to learn from those experiences. So we also wanted to see exactly what's happening in the brain and what's being disrupted. And over the years, my lab has mapped out many of the changes that are happening in the brain while birds are engaged in this learning process. And so we've identified that in HVC, structural plasticity at synapses in HVC is essential for them to form memories. Strengthening of synaptic connectivity between the neurons in HVC is also a hallmark of memory formation in this circuit, and then the subsequent changes in neural activity that ultimately underlies this pattern generation within this network. All of these things are directly impacted during these social experiences when birds are first forming memories of their dad's song. And when we looked at these in the FOXP1 knockdown birds, we found that all of these aspects of structural and synaptic plasticity and changes in how the network fires after having these social experiences, they're all disrupted and they're all blocked. And this, I think, provides us some clues as to what could be happening in the brain of kids that have disruptions in FOXP1, where they're not able to learn speech and language, and in part that could be because synaptic plasticity in neural circuits that are essential for forming these initial memories that they use to then guide subsequent learning and imitation is being disrupted. Okay, so what I've told you is that songbirds provide a powerful model to examine brain mechanisms for the cultural transmission of behaviors like speech and language. Brain circuitry used in forming social memories and learning through imitation have now been identified in this model system. And autism-linked genes also impact memory and imitation of song learning. Specifically, FOXP1 is essential for encoding song memories, and it might play a similar role in humans. So what you might be thinking to yourselves, though, particularly as I start saying things about how this might tell us something about what's going on in the human brain, is, how do avian brains relate to our brain? Is this have no relevance whatsoever, or are there some ways that we can compare these? Again, collaborating with Jenna Konopka's lab, we've been looking at the cell types in the avian brain. We've been dissecting out areas that are involved in vocal learning and vocal communication, like this area HVC, using single nucleus RNA sequencing approach to look at all of the genes that these different cell types are expressing, and the expression of those genes tend to categorize the different cell types, and to see if we can provide kind of an atlas of the cell types in the avian brain, particularly those that are involved in these interconnected neural networks that are important for this type of learning. And then we've been comparing them to the cell types that have been described and atlased in the mammalian brain. And I'm not going to go into this in any detail, but just to give you, a quick overview of what we found so far is that the neurons and these areas in the avian brain that are needed for vocal learning do not develop from the same precursors that end up making our cortex. And we think of our cortex as kind of the seat of all of the really complicated things that we do, and critical for us to learn from social experiences and to engage with each other and vocal communication and all of that. But what we find is that when we look at the effector genes in these neurons that are important for vocal learning in songbirds, is that there's strong convergence in the effector genes that they're expressing. These are the genes that are helping define, the channels that these neurons are expressing and the connections that they're making, and ultimately the kind of computations that they could be engaged in, that there's strong convergence between these networks in birds and neurons in the mammalian cortex, and specifically neurons in layers 2, 3, and 4, and 5. So, this provides us some hope that what we're learning in songbirds is going to provide some insights into what ultimately could be happening in humans, in mammals in general, when these genes that cause neurodevelopmental disorders are disrupted. Okay, and so just to finish, I'd like to thank all of you for attending, either virtually or in person today. I'd really like to thank Jennifer Holdway, my wife, who's also helped run my lab since I started here at UT Southwestern, and my entire lab that shows amazing dedication and profound interest in the type of stuff that we're doing, day in and day out, working really hard. It's a really, really great group and dynamic group. And of course, I'd also like to thank the UT Southwestern administration for supporting us. This is really a great place to run a lab, and I've felt greatly supported while I've been here. And the ARC for taking excellent care of our birds all these years, and of course the Department of Neuroscience and Joe Takahashi, and also the O'Donnell Brain Institute and Bill Dower for all the support and encouragement they've showed over the years. Thank you very much, and I'm happy to take questions.
Thanks, Todd, for a truly interesting and thought-provoking presentation. We're going to open it up for questions just to those who are online. If you put your questions in the Zoom or the chat on Zoom, Dr. Lisa Dennison is here to be your voice and raise those questions. So with that, let's open it up for questions. Over here.
Hey there, Mark Courtney from Emergency Medicine. Thank you very much. This was fascinating as somebody who really loves birds and also kind of the science of learning. My question is, you've described incredibly detailed sensory and motor feedback loops for the males, learning from males. On the other hand, we know that the females need to be able to recognize the song and have a significant amount of accuracy detection as well. So is there something to be learned from how the females learn the song or the female brain? So what might be there for opportunities for learning?
Yeah, I think that's a fantastic question, and something that we're starting to look at. It's been relatively less well studied, but some people have studied this in birds where both the males and the females sing, and there's many species of birds where that is the case, and they'll duet back and forth. But these song circuits that we studied, they exist in both males and females. And we think that the same circuits are engaged in learning when the female is also listening to their dad. You know, the video I showed you is kind of a laboratory contrived situation where we've got just one young bird and we've got one adult and they're being tutored. But how they actually do this learning is there's a clutch of birds that has both male and females, and the dad's singing to all of them, and both the males and the females are learning from listening to their dad. And there's a lot of behavioral evidence that the females are using their dad's song as a reference point to understand whether or not an adult male is going to be a suitable partner. And so their dad does, play a vital role in how they're going to be choosing males when they're older.
Todd, I'm going to jump in for the next question. You showed on the spectrograms at least a very close similarity between the young bird and the dad. How precise is that? Does the song actually evolve? Is there, sort of personality that comes in? Are there virtuoso finches? And if you look like generation after generation, is it a consistently reproduced, or does this proliferate into somebody's unique song?
So yeah, there's a couple answers to that. So first, in situations where we're just raising a male that's being tutored by just one bird, and then he's alone in his chamber and not interacting with other birds, he will, with very high fidelity, copy that song. But if he's being raised in a much more normal situation where he's got siblings that are also learning dad's song, what happens is they tend to all learn dad's song, and then they move away from each other near adulthood. So each one of their songs has its own signature. But us as experimenters, we can look at those songs and we can say, they learned their dad's song, but they all have subtle uniqueness to it. And these songs will perpetuate from generation to generation if it's a good song. Now, if dad has a bad song,
it's a dead end.
Yeah, if dad has a bad song, interestingly, what happens is that the young birds improve on dad's song, and over consecutive generations, they will get that song. So you could take one of these birds that had to improvise its song, and in a laboratory, in the wild, that bird would never mate. But in a laboratory, we can put them with a female, and they will eventually mate and have offspring. And his offspring will learn his song, but they'll improve upon it. And then you take those birds and make them the fathers for the next generation. You do this consecutively over 4 or 5 generations. And eventually those birds get back to wild type. So they get back to the point where they're just as good as any other zebra finch.
Other questions? Oh, yes, Mark.
Can you hear me? Okay, thank you. A lot of the studies you've done, for obvious reasons, are loss of function in the learning or the memory.
Yeah.
And I'm wondering about the opposite. So are there centers that can increase the acquisition of longer sequences? And along those lines, is it possible to exploit the genetics in the birds to find, sort of by comparing those who propagate short, inferior sequences with superior versus longer ones?
So we haven't identified centers, where we can manipulate the activity and make the behavior better. In part, I think, is because there's likely ceiling effects for this network. One of the things that we're doing is collaborating with other labs now that are studying songbirds that have much more complex songs, like European starlings, which can sing for minutes without repeating themselves. And we want to look in detail at this circuit HVC and see, because both of these birds have the exact same brain circuits, but one of them shows really remarkable complexity and the other one not so much. And we want to see what are the underlying differences between these networks, in the pattern generation circuits and how they're connected up, to try to understand, how evolution is tweaking these networks in order to enhance the ability to learn things.
Okay, we're going to do 3 quick questions before we're kind of out of our established time, but it's a great selling point for our reception, which will be on D1 just as soon as we do. We'll take one from Zoom and then a couple more from here in the Gooch.
Quick question from Zoom. How did the female birds feel about the opto-tutored birds?
They're not impressed. Yeah. The females, like I said, they listen to their dad's song and they use that as a reference point, and these opto-tutored birds, their songs are highly abnormal.
Okay, the question, yes.
Thanks. Steve Small from the Department of Neurology. I study neurobiology of language, and the word language has come up a few times. You have really impressive work in vocal learning and vocal memory. I've been wondering for a long time about the relation between this and language. These birds learn a single song or multiple songs? And secondly, do they change songs for different purposes in different contexts?
Yeah, so these birds learn a single song, but they do change their song in terms of how they sing it for different contexts. And so when a male is singing to a female, he sings it in a more precise, stereotyped way, and sometimes slightly faster. When he's singing to his offspring, he exaggerates certain features that we think is kind of a form of baby talk. And then when he's singing it just by himself, when he's just practicing, he sings it in another slightly different mode. But saying that, it's all the same song. Right? And so, unless you're doing quantification of these things, you would never be able to tell as a human observer looking at it or listening to it. But the females, for example, know instantly whether or not the male is singing his practice song or his courtship song. And they can detect that very, very easily. But in terms of, this is just one species of bird, and we really focus on this species because their song is simple. And it's more tractable for us to study in a lab. When you have an animal that can learn from lots of different auditory experiences and learn lots of different things, controlling that in a laboratory environment and really being able to prove that this is what you're dissecting out and learning from it becomes much more difficult.
Okay, last question, the young man in the first row.
What if a Bob Dylan or Willie Nelson type singer comes along that the birds have never heard of before? Can he ultimately influence the flock, or does he get ostracized?
So there are I don't know about being ostracized or not, but we can raise these birds with other species that have more complicated songs and see how they learn from those more complicated songs. And what we find is that as long as the species is close enough in terms of the spectral and temporal properties of the behavior, they will copy the syllables of the other species. And They will ultimately, though, not match the complexity of the other species. So in birds, that can change things up a lot in how they're singing. These guys won't do that. They'll learn the syllables, but then they'll sing it in a very stereotyped way and not, be able to do all sorts of complicated things with it.
So if you ever do get to Willie Nelson, I think I know we can get a volunteer to help you finish the song. Well, thanks, Todd. Really terrific. And as I said before, I really hope everybody will take the time to join Todd down on D1. And on your way out, also to learn more about the Equally Able Alliance, or BRG. Delighted that you're here. Just so you get it on your calendar, our next PLS will be on February 13th. Dan Siegert from Bioengineering. We'll be talking about his work, creative and innovative ways of applying nanotechnology to delivering genetic packages to very specific tissues and with a specific goal in mind. So I'm sure that will too be a great presentation. But Todd, thanks very much for a great afternoon.